Welcome to Biosumit friends. Today we learn about genetic recombination in bacteria. Now, what is genetic recombination? Actually, it is the exchange of genes between two DNA molecules to form a new combination of genes on a chromosome. Right? So, why it is needed? It actually contributes to a population's genetic diversity. And diversity, the variations. Right? Now, this source of variation actually leads to evolution, and to survive, evolution is very much needed, right? Now, there are two types of methods of transmission of genes. One is vertical, one is horizontal. The vertical gene transfer, that happens in plants and animals, right? Like the genes are passed from an organism to its offspring, or rather to say, it transfers or it is transferred, rather to say, from one generation to another generation, right? This is vertical transmission or vertical gene transfer. But for horizontal gene transfer, as I have written here, you can see this actually takes place in bacteria. How? It actually passes the DNA laterally to other bacteria. It doesn't need to transmit their gene from one generation to another generation. That's why it is called horizontal, right? Now, to accomplish this, it will involve one donor and one recipient cell. Now, donor cell will give the portion of DNA to the recipient cell. Okay, the recipient cell is going to receive the DNA from the donor. So here the mechanism works, right? Now check this diagram. Here I have tried to make you understood through this schematic diagram how the recombination works. Okay, so here you can see this is D and this is R. D means donor and R means recombinant. So this is the donor cell, or you can say donor DNA, and this is the recipient DNA. Okay, and here you can see there is a nick in this donor part, right? Nick means there is a cut in one strand of the DNA. Not cut actually. Cut is said when the this nick will be in the two strand. Let me speak in easily. When this cut will be in the two both the strands, it will called simply as cut. Okay, and if it's in one strand, then it can be called nick. So here you can see from here, okay, this is going to be cut and it's going to be merged here, but it's not the thing. Like it can't join any part of this recipient, right? So what happens, DNA from the donor, this only aligns with the complementary base pairs of the recipient. Okay, so here the base pair will be complementary, that's why it can align with this, with the recipient, right? Now this can involve thousands of base pairs, but remember here one protein will be there, okay, that actually catalyzes the joining of two strand. Okay, so this protein is called recombination A or RAK protein. Okay, so it will catalyze the joining of the two strand, right? Now in the next step you can see the part of recipient is here, okay, and the part of donor is there. Okay, this is how the recombination is working, right? So now let's now check the at first it looks like this, right? Now it is consisting some parts of the R part, and the R part is consisting some part of the D part. Now we can call it that it is a recombinant. Okay, these complementary base pairs between the two strands that will be resolved by DNA polymerase and ligase. Okay, so that's why I have written here the DNA polymerase and ligase work here. Okay, now the recipient in these cases can have one or more new genes. Okay, here I have just drawn a simple diagram to make you understood, but it can be with so many new genes for the recipient. Okay, now this is the general idea of genetic recombination in bacteria. I mean how the recombination works. Okay, now there are so many processes through which bacteria actually performs horizontal gene transfer, like, or namely you can say transduction, transformation, or conjugation. So in our next video, we'll try to understand that processes. Okay, hope this video is helpful to make you understood the genetic recombination of bacteria, the basic idea of this genetic recombination in bacteria. So see you in the next video with some specific process. Bye bye.